I'm Irv Zucker, and I'm here to interview Steve Glickman. Steve and I have been colleagues and friends for 50 years, and I have been a witness to his accomplishments throughout all that time. So it is a pleasure for me today to revisit them for a broader audience. I'm going to start, Steve, by asking you where you were born and where you grew up, what your parents' occupations were, and how the environment they created shaped you.
So I was born in 1933 in the Bronx, New York, and lived there for the first 10 years. Both my parents were teachers. My mother taught the piano at the Juilliard and also later privately in our house, but that was much later. My father went to school every day where he taught junior high school mathematics. And because they were out of the house so much, there was a woman who took care of me. We lived in the Bronx near the Bronx Zoo. And this woman's brother was the keeper at the rhinoceros house at the Bronx Zoo. And so we would go over there every day for lunch. And she would go in and visit with her brother, and I would be outside staring at this rhinoceros who I was sure was communicating with me. And that began a life of connecting to animals that persisted to this day.
Was there something in the environment that your parents created that had a lasting impact on you, other than fortuitously giving you exposure to animals?
My parents actually did not particularly like animals, so that was maybe a rebellious thing on my part. But what my parents did convey was a sense of the importance of teaching. And I certainly inherited that from both of them, that teaching was an important and good activity.
So I guess the fact that you were exposed to the zoo explains why a city kid like you ended up spending so much of his time chasing after wild animals in Africa, and you and your students did so much field work. It's not the usual thing one associates with somebody with your background. You first came to Berkeley as a Miller Fellow in 1962 under the mentorship of Frank Beach. What kind of empirical and scholarly work did you do during this fellowship, and how did it affect your subsequent research?
Well, I did two kinds of work on that fellowship, 1962 to 64. And the project that I actually began working on as a research project in Berkeley involved a group of striped skunks. And I was able to move this set of skunks into the field station in the Berkeley Hills and study their social interactions and their threat patterns and things like that. They were descented, so it was safe to approach them and do those things. And I had begun working with skunks when I was a child of 9 in a children's camp in the Catskill Mountains. And in the Camp Locanda newspaper, they wrote, "If he leaves his group and doesn't come back, he's feeding his skunks at the nature shack." And so the first thing I did on the Miller Fellowship was arrange for this group of skunks to become my friends. And Bernie Schiff, who you know very well, came out there as my graduate student and helped me with the project. And then at the end of the project, 1964, we released all the skunks in Tilden Park. where their descendants are probably still
spraying animals.
Well, yes, and having a good time. I consider Tilden Park to be a good habitat for skunks.
Okay, good. Did this 2-year interval in Berkeley somehow stimulate you to want to come back later on as a faculty member?
It certainly did. Now, I forgot to mention the other primary activity during those years, and that was writing up a set of papers on the research I'd been doing at the Lincoln Park Zoo in Chicago, where I'd been studying curiosity in zoo animals. And I began working there in 1959 when a man named Marlin Perkins was the director, and I had a grant from the National Science Foundation. And Marlin, and I used to hire the curators who worked for Marlin to help me at night after the zoo was closed to the public, when I would be testing the animals and throwing novel objects into their cages. And after watching us for about 6 months, Marlin gave us the keys to the zoo and every animal's cage. No one has ever had that kind of access, I now appreciate. But when I arrived in Berkeley in 1962, I had a lot of data to write up. And a good chunk of my Miller Fellowship was spent either researching with skunks or writing papers about data we collected at the Lincoln Park Zoo.
Can you say a little bit about how you study curiosity? You just threw the same objects into the cages of how many different species?
It was probably 100 different species. I tested every animal at the zoo where I could retrieve the objects after 6 minutes and try new objects. So I was looking for a general response to objects, not a specific curiosity, oh, I'm interested in that. I wanted a more general thing. So we used 5 different objects that were scaled to the, roughly scaled to the handling capacity of the individual animals, where they could pick them up in their mouth and bat them with their paws, or if you're a primate, pick it up in your paw and see what was the object was, why the object was interesting. So I tested virtually everything there, including, and I tested both mammals and reptiles. I didn't formally test birds, but I did some informal observations on birds. They're not in any of the papers.
And what would be one main generalization that you concluded from this investigation?
That animal curiosity in terms of investigating novel objects is very much tied to the dietary habits of animals in nature. So if you had a very narrow diet that was where the food was easily available for consumption, you were a animal that just ate grass and it didn't require any manipulation, and all you ate was grass, you wouldn't be a very curious animal in terms of novel object tests. But if you had a varied diet that required a lot of manipulatory behavior, you would be intensely curious, and you would do all kinds of interesting things with the objects, and you also tended to have a bigger brain. And that particularly was illustrated by the behavior of baboons and other very fancy primates.
It's very interesting. You taught at Northwestern and at Michigan before coming to Cal as a faculty member in 1968. How were you affected by the tumultuous conditions on campus for the next few years? And were you challenged in any particular way by students?
remember that the first couple of years I was on the Cal campus, there were helicopters overhead at Sproul Plaza, but and that was entertaining. But to be honest, none of that stuff affected either my research or my teaching, and the teaching just went along.
Okay, I was less fortunate, having been tear gassed twice on campus during that time. You received the Campus Distinguished Teaching Award. How would you describe your teaching style in large lecture classes and in smaller seminars?
Well, I never had a theory of teaching. When I watch the current recipients of these teaching awards get up and talk about their teaching, they've obviously thought about it in very interesting and personal ways. I just got up and taught. And later on, people like you and my students in their student evaluations pointed out to me that what I was doing was telling stories. And when I was teaching history of psychology, I would make a point of telling stories about the backgrounds of the different famous psychologists that I was talking about, to get myself and the students involved in the lives and intellectual productions of these people and how their lives related to their intellectual productions. So when I taught history of psychology, I didn't go back to the ancient Greeks. I started somewhere around the time that, about the beginning of the 19th century, when people were first beginning to understand the nervous system. And so I talked about the lives of the Volta and Galvani and all these people, leading up to people like William James, who I spent a lot of time on, and also Sigmund Freud, who I spent a lot of time on.
I seem to recall that you were asked by a group of students to run a seminar on sickle cell anemia. I didn't think that that was one of your areas of expertise. What was that all about?
Well, sometime in the early 1980s, a group of black students approached me and asked me to give them a seminar on sickle cell anemia. And I explained to them that I didn't know anything about sickle cell anemia. And one of them said to me, "Let me understand this. You can only teach things that you studied in graduate school and are totally familiar with." So I said, okay, look, I can learn about sickle cell anemia and then I can give you a 1-hour lecture. But that's not what I could really teach you. And that's not a full course. So if you want to take a course with me, we can create something in which we will review the at that time the Jensen studies on heredity and IQ, and we can figure out what's wrong with them. And also, you could do research yourselves if you come up with some ideas, and they did. And it was a wonderful seminar. And they had developed this hypothesis that residents who lived in low-rise projects like Acorn would be much more content and in their residence if they knew their neighbors in low-density projects, as opposed to the people who lived in high-rise projects in San Francisco. And they developed a questionnaire, and then they went out and got data on the relative satisfaction with their living conditions of people in low-rise and high-rise projects. And it was a beautiful study that was in many ways ahead of its time. And it couldn't have been done by white faculty members. It required black students who could go in and, or white students. It required black students who could go into these projects and approach the people who were living there and get good, honest answers. And I admired it so much. And that was perhaps the most memorable seminar I ever taught, and I followed the careers of several of the students subsequently. One of them went on to get a PhD at Stanford in psychology. Another went on to law school and became a serious lawyer in Los Angeles. And it was a happy outcome in every way.
You mentioned about the fact that you taught the history of psychology. I believe you started that course in the local department. Isn't that correct?
Yeah, I can't remember.
Okay.
Which is not an uncommon experience for me at this age.
But it brings me to two related things, which is more generally, which courses engaged you, and why do you think you emphasized historical antecedents in your courses more than other faculty did?
So when I came here as a faculty member in 1968, my assignment was, at the undergraduate level, was to teach physiological psychology, as we called it in those days. And I enjoyed that. But I enjoyed the next course I taught a bit more, which was animal behavior. And we also had an interdisciplinary version of animal behavior, which was taught with colleagues in zoology. And that was a nice course, and I enjoyed that. But then when Lucy Jacobs joined our department and was ready to teach animal behavior, I said, fine, Lucy, you teach animal behavior and I'll teach history of psychology, which has always been an interesting topic for me. And it was just a gift to be able to learn all that stuff and talk about the people and recreate the history of psychology in my own style.
I sat in on that course the very last time you taught it, and it was remarkable for the breadth of the coverage, but I was also amused by the fact I sat in the back of the room, and in front of me were students on their laptops. Some of them were playing video games, and you were writing on the board at a time when most instructors were using PowerPoint presentations. You have any thoughts about that?
Well, I'm glad that I wasn't focused on those students that were playing video games.
I wanted to yell at them.
But there were enough students who were paying attention that I would make eye contact with them. And several students came up to me and said, when we saw you writing on the blackboard, we knew we wanted to be in this class.
Oh.
So there were other rewarding parts of my antiquated mode of instruction. And then, as you know, at the end of the class, the last class, a group of students from the class baked a cake and presented it to me at the end of the class, and it was a chocolate cake with a hyena on it. And I'm supposed to be riding the hyena. And I've given a picture of that to the people who are filming this.
Excellent. Good. Can you say a little bit about your committee assignments at the university? You were there for a very long period of time. You must have done some committee work.
Yes, I was on the original Title IX committee, and that was an interesting experience, and glad to see Title IX implemented at Berkeley. And then the other really interesting committee was the Budget Committee, where I had the pleasure of sitting and learning about all the wonderful research that goes on at the University of California, Berkeley, from colleagues in classics and departments that I knew nothing about. And
can you say something about what the mandate of the Budget Committee is?
Well, the Budget Committee at Berkeley reviews every pay raise, appointment to the faculty, and promotion, and makes recommendations to the administration. And it was an interesting job, and I just had a very good time. And I also, not everyone works out on the Budget Committee, and you have to be able to get into the social dynamic. And I met people on there in history and English and other areas who are friends to this day.
Wonderful. You also served 5 years as chair of the psychology department. And I'm wondering what your greatest challenge was during those 5 years, and with hindsight, your most satisfying achievement.
Well, I'm not sure how to answer that. The greatest challenge was to get a group of 30 or 40 faculty, each of whom had their own domain of research, to cluster around the common theme of psychiatry, to regard themselves as members of a unified department. And I tried various strategies to get that happen, and including inviting guest lecturers like Janet Taylor Spence, whose work was later president of the APA, and I tried to get people like that in as lecturers. Also, I tried to lecturers that would attract the whole department. And I also worked on the issue of getting more minority psychology taught in the department with the help of people like Phil and Carolyn Cowan. And that's where we created a new seminar, and we invited this guy, Robert Guthrie, up to lecture he'd been in Florida and then he was in San Diego. And Robert had written a book called Even the Rat Was White. And he came in and gave a wonderful lecture. And then other local people came in and lectured, and it was the first course in that kind of diverse focus. And I felt good about that.
Was it during your tenure as chair that women finally got to become regular faculty members?
Well, they've been appointed when I got in, and at least 3 of them received tenure while I was chair of the department, and I prepared their cases. Now, the credit for that goes to the women, not to me. They were the ones who did the work. But it was a good feeling, and I remember 2 of them got tenure on the same day, and the provost stopped me on campus, told me about their promotion to tenure and congratulated me. And we talked about congratulating them. And that was a very rewarding part of being chair of the department.
And you were also the director of the field station for animal behavior for something like 14 years. You succeeded the founding director, if I recall correctly. Was that a fun job?
Not as much fun. I think that we did have some nice projects there, and diverse projects, and the idea in that field station, as created by Frank Beach, was to provide semi-natural habitats for animals so that they could have a life that was richer than that experienced by most animals at the university who lived in laboratory cages. And it would be a semi-natural environment for many of them, and it was fun doing that. Lovely work was done with birds and birdsong following that arrangement, and as you know, eventually I was to introduce hyenas to that.
We'll get to that shortly.
Okay.
You have had a long-standing interest in the 19th century biologist Alfred Russel Wallace, who is the co-formulator of the theory of evolution through natural selection. Why Wallace rather than Darwin?
Because when I read about Wallace, I loved how this poor, self-educated boy in England had taught himself biology and made himself into a very serious biologist. He worked as a surveyor for his brother, who was in the building trades in England in the 1830s and 40s. And in the late 40s, on his own, Wallace just takes off for South America, spends 4 years there studying animals, then comes back, and a few years later goes off to the Malay Archipelago and spends 8 years in the Malay Archipelago. And while he's in the Malay Archipelago, in 1855, he gets the idea for the transmutation of species, a mechanism, and the mechanism is natural selection. The contrast with Darwin becomes particularly dramatic when you compare. I mean, Darwin and Wallace had a major disagreement, and the disagreement was over the evolution of the human brain. And Darwin was ready to see these peoples who lived in the jungles of the world as halfway as smart as Englishmen. And he actually has some comments like that in his own writings about their moral qualities. And Wallace lived with these people wherever he went. He wasn't rich. He didn't live surrounded by Englishmen. He lived with the people in South America.
What was his view?
His view was expressed in the last pages of the Malay Archipelago book. And he said, you know, these people have such high moral standards, and that each of them respects the right of his neighbor. And he contrasted that with the situation in England, where you have to have all these lawyers and police, and there are also so many paupers. The percentage of the English population that were paupers and in trouble with the law and so forth. And so he thought that these people were every bit the moral superiors of the average Englishman, unlike Darwin. And he also thought that they were remarkably smart. And he made a dictionary of their languages while he was out in these places. And and he thought that they were as smart as everybody in England, and it didn't make sense to him that this could have occurred through natural selection, because he thought that their daily life did not require the kind of things that they were capable of doing if they'd been sitting there in England. And he was wrong about that. Life was pretty challenging out there, and they knew a lot more than he understood. But he decided that the human brain had been created by an unnatural force, a supernatural force. And he began attending seances and decided that there were forces beyond those we were seeing on Earth, and that we all would have eternal life when we were reincarnated. And it was he went on from there. But you were going to ask me, I think, about the trip that Krista and I did.
That's a good place to go.
And in 1997, Krista and I traveled around Wallace's Indonesia and out to some fairly remote places, like the island of Ternate, where he composed his theory of natural selection paper, which he sent to Darwin, by the way. And because Darwin was the only biologist he knew was working on the problem. And it was fascinating to go around to these places and see what could still be seen. And a lot of it could still be seen, and that's what we were looking at on Ambon and Sulawesi and all kinds of remote, to us, places.
You're most identified as the leader of an unusual long-term project investigating the biology of the spotted hyena, primarily in a colony that you founded in the Berkeley Hills above campus, adjacent to Tilden Park. Can you say something about how you got involved in this enterprise? And what is unusual about this species that drew you to it?
Well, in the fall of 1982, two old friends, Lawrence Frank, who was a graduate of the PhD program in zoology and had been working with hyenas in Kenya, and Julian Davidson, who I'd been a postdoc with in Berkeley and who was an endocrinologist at Stanford, approached me at Shattuck Avenue Spats. They invited me to lunch, and they had a proposal. They had been working on some peculiarities of hyenas in the field, but had reached some limits, and I'll have to tell you what those peculiarities of hyenas were. And they thought hyenas needed to be brought into captivity. And so, because I was working at the behavior station, they thought maybe I could arrange it. And then they also knew that I'd spent some time working with strange animals. And so they invited me and Krista over to Africa to see Lawrence's field site. And I came home and I told Krista that I'd been invited to do this, but I was just completing my 5-year term as department chair, and I was worried that my research career was in a shambles and I needed to really get things going, and this work with hyenas was so crazy that it was never really going to happen. And Krista said to me, let me understand this. Lawrence Frank has invited us to Africa to live in a tent on the banks of the Talek River, surrounded by elephants and zebra and giraffes and hyenas, and you don't know whether we should go. Why are we married? And I capitulated. And in January of 1983, Krista and I went to Africa, saw all the things that Lawrence wanted us to see, and I came back and spent the rest of the spring writing a grant, which was submitted in July of 1983 and ultimately funded in July of 1984. And I thought it was pretty astounding that we got funded, given that we were proposing to bring over squads of baby hyenas from Africa, transport them 9,000 miles to Berkeley, California, have them grow up in captivity and behave like hyenas in Africa, and reproduce. And it was we hadn't shown that we could do any of those things. And one could have reasonably been very skeptical, but for some astounding reason, a grant committee gave us money. And then it worked. Partly thanks to the incredible arrangements that Lawrence Frank made in Africa, coordinating with the local Maasai, who are essential to collecting hyenas. Now, the Maasai culture is very much involved with cattle and sheep, and hyenas were their enemies. And they were poisoning hyenas in the area where we collected them. So the Maasai were happy to help us. And they said, how come you're only taking 20? Take them all. But we only took 20 and brought over 10 each year over two years, and we designed a facility in Berkeley that could house them. And housed them there, and they grew up and reproduced. And why they're interesting now has to be talked about. Okay?
Good idea.
Okay. So hyenas are social carnivores that live in clans. Clans where they have matrilines like many old world primates. And they're very active hunters. By hunting in groups, they can bring down prey as large as buffalo and zebra. And they're not just scavengers, although certainly scavenge, but in one part of the Serengeti, Hans Crook reported that 60% of the lion food was originally killed by hyenas. And then the lions drove the hyenas off to kill, which drives them crazy. And the lions scavenged. So they were interesting because of, and within these clans, every adult female totally dominates every adult male. The females are much more aggressive than the males. And the males are ordinarily terrified of the females. Now, as long as they stay in the clan of their birth, they have their mother's rank, and they're ahead of all the immigrant males. But if they're going to reproduce, they're going to have to leave the clan of their birth, go elsewhere to mate and join a new clan. And that's a tough process and takes a lot of years before they rise in the male hierarchy. So it's a fascinating social system. And we were going to study all their behavior in captivity and how it developed. But then there's another characteristic of hyenas that had attracted attention since antiquity and had been written about by Aristotle. And that is that the genitalia of female spotted hyenas, not brown hyenas or striped hyenas, but spotted hyenas, have external genitalia that to the casual observer are not distinguishable from those of the male. They have a scrotum, like a male, instead of an external vaginal opening. The clitoris has developed till it looks just like a penis. They get erections like males. And they use those erections actually when they say hello to one another. The hyena meeting ceremony, the subordinate animal gets an erection and comes up and offers it for inspection by the dominant animal. And we were going to study that in captivity too. Anyway, this extreme masculinization of the... Oh, and I have to... There's one other thing. Because I have no external distal vaginal opening, the clitoris is also traversed by a central urogenital canal. And hyenas mate and ultimately give birth through the tip of this large clitoris to a 2 to 3 pound baby. So the clitoris expands and ultimately babies pop out of there. And they've mated through there to get in that condition. And the whole thing just seems so unreal. And it still is amazing to me. And it's not a very good way to do things ordinarily. A large number of the first births are stillborn because the babies get stuck in the clitoris and can't get out, and the umbilical cord's detached. And if you don't come out in a timely manner, you die of asphyxiation. So the whole system seems so strange. And Lawrence and Julian had begun studying the mechanism that produced the genitalia in Africa. And it's to this day, the only theory of why males have male genitalia and females have female genitalia involves the early action of male hormones or androgens at critical stages in development in our species and most mammalian species, in utero. So that had to be, if the current theory of how you made male genitalia was true, these females had to have high levels of androgens circulating at this critical stages of development, unlike all other female mammals. Spotted hyenas are the only mammals on Earth that have this arrangement. And Julian and Lawrence had been looking for androgens in nature and found out there are limitations to what they could do out there. So at Shattuck Avenue Spats in the fall of 1982, they approached me about bringing some to Berkeley so we could really figure out how this system worked. And the Berkeley Hyena Project began, and something happened pretty early on that was going to change the nature of the project. And what happened was that one of my colleagues, Julian Davidson, developed early Alzheimer's disease and had to close his laboratory. But before Julian did that, he arranged for a faculty member at the University of California, San Francisco, who is an endocrinologist in obstetrics and gynecology, to join our project as an endocrinologist. And his name was Pentti Finn Sideri. And that changed the whole nature of the project, because Finn, who had worked on these problems of sexual differentiation, said, you know, Steve, we need a real anatomist to look at the details of the clitoris and the penis. And he recruited a guy named Jerry Cunha, who was in anatomy at the medical school. And then Jerry began working with us, and he said, Steve, there are human medical conditions where little girls are born with male genitalia. And that is studied in the Department of Urology. And so Jerry recruited Larry Baskin, who's the chief of pediatric urology at the medical school. And then Finn also recruited a man named Gene Wilson, who's one of the fathers of the field of sexual differentiation. And Gene went and found someone at the University of Texas Southwest Medical School, Mike McFall, who was a molecular endocrinologist and worked on the molecular endocrinology of the Hyena androgen receptor. So a bunch of things now began to happen. First, my wonderful colleague at Berkeley, I needed an endocrinologist in Berkeley. I didn't know anything about this. I'd entered this field of sexual differentiation at the age of 50 with the level of knowledge that one would have in an introductory textbook. I knew nothing about them. But talking to all these people now was beginning to educate me, and I learned that, gee, all endocrinologists aren't alike. And so I began recruiting other endocrinologists. First, Paul Licht at Berkeley to replace Julian as the primary endocrinologist on the project. And then people like Al Conley at UC Davis, who was an expert in the enzymatic transformation of one hormone into another hormone, androgens to estrogens and so forth and so on. And gradually the network began to build up. An expert in relaxin, a hormone that relaxes female tissues to promote the birth process in humans. He read about the hyenas in a story in the New York Times, and he wrote to me and was interested in studying relaxin in hyenas. So little by little, we built up this big network of collaborators on the Berkeley Hyena project. And it included people you know, Geert de Vries and Nancy Forger, two of our friends, who came in as the primary nervous system people to look at the hyena nervous system and ultimately published things on hyena aggression from a small number of brains that we managed to collect largely at a special expedition to Moyali in 1990.
So would you say it's fair to characterize you as the conductor of an orchestra made up of many, many people from different disciplines, most of them not in Berkeley, some of them in other countries, and you managed all this for more than 20 years.
Yeah, well, the hyenas were here for 30 years, and I managed to keep funding going for about 30 years, although that was always a challenge.
So were some people in the medical establishment interested in what you were finding, and did they come and visit?
They did more than visit. For example, this fellow Larry Baskin got very interested in the maintenance of sensitivity in the human clitoris, and had been one of the first two people to map the distribution of nerves in the human clitoris, and they're on the dorsal surface right off the midline. But some of their clitoral surgeries involve women who've been exposed to high levels of androgen in utero and have a clitoris that is the size of a penis. And so they have to go in there. If these people are going to have a normal sexual social life, they have to go in there and do clitoral surgery. And while they're doing it, Larry wanted to make sure they preserve the sensitivity of the clitoris. And so he on his own, he did a study with hyenas where I gave him some hyena genitalia, and he maps the peripheral nerves in the hyena clitoris as an animal model of the human hypertrophy clitoris. And indeed, the nerves are in the same location pretty much in the hyena. They just go further down toward the end. So that is now an animal model that is referred to in the human medical literature. And another case, there was a little girl born at the medical school with male genitalia. And they'd been following her mother's hormone levels during pregnancy, and an awful lot of androgen was in there that shouldn't have been there. And it turned out, when they looked, that ordinarily in women, there's a lot of aromatase, the aromatase enzyme in the placenta, and it takes any androgens and it makes them into estrogens, and they can't masculinize the developing fetus. But in this woman, there had been a mutation, and her enzyme wasn't working properly. And so she developed very high levels of androgen, and we had previously found that in the hyena, one of the main sources of androgens at critical stage development is the placenta. There are all those enzymes in there making androgens. And what had happened in this woman was that her placenta was behaving like a hyena placenta and was masculinizing her daughter. And in the paper that my colleagues at UCSF published about this case, they refer to the hyena and our work on the hyena placenta by way of understanding what happened in that woman. So you had basic science proceeding along its track, but because of all these collaborators with different expertise, a network developed which translated basic science into useful medical discoveries.
Neat. So I want to switch gears now and ask you, when did you formally retire from teaching and committee work?
Again. I retired at the age of 75 in 2008.
Okay, and did the Hyena Project continue post-retirement?
Yes, the last hyena left Berkeley in December 2014.
So 6 years after you formally retired.
Yeah, and I should say something else about that, because I'm still writing papers about hyenas, but we might have been able to keep the project going beyond that. But a likely way to go would have involved euthanizing the hyenas and studying their nervous systems. And they were all our friends, and we couldn't do that. So we decided to shut the project down.
Excuse me? What happened to the hyenas?
And all the hyenas went to good homes in zoos and animal refuges. We were actually the single largest supplier of hyenas to zoos around the world. If you go to Paris, you can see spotted hyenas at the Paris Zoo that were...
Are you saying that you can't order a hyena from Amazon?
Not yet.
Next
year.
Okay.
And the woman whom, I should mention the woman who managed the Hyena Project from its inception and arranged this network of collaborators and arranged all the meetings and everything, and it ultimately arranged to give all the hyenas away, and that's Mary Welderly, who was
So I have
really
two more
incredible.
questions I want to pose to you. The first one is, could you have foreseen your scientific trajectory when you completed your graduate work?
Not at all. When I left graduate school at McGill, and I was trained in
studying brain and behavior and doing studies in lab rats, on the lab rat brain. I didn't know where I was going to go, but teaching at Northwestern and living near the Lincoln Park Zoo, I began to go around the zoo.
Okay.
As a hobby. And one day I just knocked on Marlon Perkins' door. I knew I really wanted to get in there and play with these baby snow leopards and baby tigers. And I knocked on Marlon Perkins' door and I got in to talk to him. And I proposed studying color vision in big cats. We won't go into the reasons for that, but I never did it. Instead, once I got in there, I began studying all these other things. I did study color vision in brown lemur at one point, and brown lemurs at one point, but it was just a playground for me.
Okay, and the last thing I'm going to ask you is, if you were going to do it all over again, what would you do differently and what wouldn't you change?
I'm not sure that I would do anything differently. It was all, I feel I've led a privileged life. Sometimes it just amazes me that the people of California paid my salary all those years when I was just going over to the university and doing things that I love to do, whether it was standing up in front of a class and telling them about historical figures that they'd never wanted to learn about. One of the nicest things that I would get in student evaluations, one of the things that warmed my heart the most, was when students would write how they expected the history of psychology to be really boring. And then because I'd managed to engage them largely with stories about the lives of these psychologists, they found they enjoyed it. And I just think I've had a privileged existence.
Well, having had the privilege of teaching with you for 10 or 15 years, it was a remarkable experience to learn from these stories that were so much more accessible than a professor getting up and pontificating in technical kinds of things. And the remarkable thing was that at the end of the story, you managed to communicate all the important scientific points. As one of our senior colleagues who sat in on your course came in to say to me, he's up there talking to 300 students, telling stories, and at the end of it, he's covered all the important points, and then he paused and he said, "I can't do that." And he left. So it's been a privilege. Thank you.
And thank you, Irv. It was wonderful teaching seminars with you, and observing you was crucial to my being able to write grants that would actually attract some money, which was required to support those expensive hyenas. And you were the most successful grant getter I knew at Berkeley. And I could never write grants that were as tight as yours. But you did change me from writing "I wonder what would happen if" grants to writing grants that had well-defined questions. And I learned it all from you, and none of that stuff would have happened if we hadn't been friends. And I feel very lucky about that as well.
Thank you. I know you didn't give me any of the money that you got.
No, I was cheap.
Okay.